Now, in this set of masterclass, we are going to learn about translation. What is translation? Translation is nothing but formation of protein from the template of mRNA. That is translation. Before we learn translation, let's look at a very important concept called as cistron and monocistronic and polycistronic. So what is cistron? Cistron, by definition, is the smallest unit of code for any polypeptide synthesis. Okay. So for example, if you see this prokaryotic gene here, so this was the protein, this was the promoter sequence, and it has three cistrons, which means three proteins can be formed from that. Okay. Similarly, if you see eukaryotic gene, here is the promoter sequence, and it has only one cistron, which means every gene will be forming one protein. Now, what is basically it looks like? It looks like something like in eukaryote. Eukaryote is like, you have a separate switch for your light, you have a separate switch for your fan, and you have a separate switch for your in this set of masterclass, we are going to look at translation. So what is translation? Translation is formation of protein on the template of mRNA. That is translation. Before we look at the details of translation, let's understand a very important concept called as cistron, monocistronic and polycistronic. What is cistron? Cistron is the smallest unit of code for any polypeptide synthesis. For example, here you see this entire, length of mRNA codes for this protein. Here, if you can see, you know, there are three lengths, okay, three cistrons present in a single gene. Okay, now it is said, so these are three cistrons basically. Now it is said that eukaryotic RNA is monocistronic, which means a single, protein polypeptide will be formed from each cistron, and each cistron will have a separate promoter sequence. Okay, and when we talk about polycistronic gene, multiple genes, multiple cistrons can be under one gene, and if this and multiple cistrons can have a single promoter sequence. So if this promoter sequence is on, all these three proteins will be formed. If it is off, none of these will be produced. So let me give an example analogy so that you will understand. Let's say in your house, if you have one switch, and the moment you open that switch, your light, your fan, and your TV, all three starts opening. So that is the example of polycistronic gene, which means the moment you on the promoter, all the three will be on. If you off the promoter, all the three will be off. So that is prokaryotic mRNA. Okay, polycistronic gene. When you talk about eukaryotic, you have different switch for your fan, you have a different switch for your light, you have a different switch for your TV. So what happens if you open only, you can only open the TV switch, or you can only open the light switch or your fan switch. So that is the example of monocistronic. So this is basically a very important concept. The next important concept is around the genetic code, the concept of genetic code. Now we know that every codon is basically a triplet, which means three nitrogenous bases combined together to form a codon. Okay, so all the possible combinations of this triplet sequence is basically what we call as genetic code. For example, and this genetic code was given by Nirenberg and Harbind Khurana. So every position, at every position, if you see, so there are three positions, 1, 2, and 3. Every position there is four possible combinations. Okay, four different types of nitrogenous base can come. So the total number of combination is 4 into 4 into 4, 64. So 6 to 4 is the total number of codons. Is it clear? 64 is the total number of codons. And out of this 64, we know that 3 are stop codons, okay, and rest 61 codes for any amino acid. So this basically, this what codon will code for what amino acid, this is basically your genetic code. Okay, is this clear? For example, in this particular slide, you can see that UUU. It's basically a codon, and this codes for phenylalanine. For example, UUC, again it's a codon, again it codes for phenylalanine. So this is your genetic code. As I told you, there are four types of bases in our body. There are 20 amino acids. So 64 codon can include 20 amino acid, but we know that out of this 64, 3 is a stop codon. So 61 codons will code for 20 amino acid. So what are the stop codons? The stop codons are UAA, UAG, and UGA. So this has been asked multiple times in examination, also called as ochre, amber, and opal. Okay, ochre, amber, and ochre. So UAG, UGA, and UAA. These are the stop codons. Now there are certain various characteristic feature of this genetic code. Our genetic code, there are some characteristic features. And again, questions are asked on the characteristics of genetic code. So let's look at, the characteristic of genetic code. Before that, let's see one important point, that stop codons generally do not code for any amino acid. However, UGA and UAG. UAG will code for pyrrolysine and UGA will code for selenocysteine by a process called as recoding. I've already taught this recoding in your amino acid metabolism classes. So remember, and then UGA in mitochondrial DNA will code for tryptophan. So these are three exceptions of the stop codon. Start codon, always AUG is the start codon. So it represents methionine in eukaryote and N-formyl methionine in prokaryotes. Okay. So this is the about the stop codon. Let's look at the various features of genetic code, because this is very important. The first feature is triplet codon, which means every codon is made up of three nitrogenous bases. For example, U or UUA. So this is the first. Second is about terminator codon. We have talked about stop codon. Initiator codon. We have talked about AOG, which is our initiator codon. Genetic code is degenerate or redundant. What do you mean by degenerate or redundant? Now, if you see, total 61 codons are there, okay, and only 20 amino acids are there. So this means that more than one codon will code for the same amino acid. Okay, for example, UUU and UUC both code for phenylalanine. Okay, so more than one codon codes for the same amino acid. This is called as degeneracy or redundancy. Again, this is a feature of our genetic code. It is unambiguous. What do you mean by unambiguous? Which means even though it is degenerate, okay, even though it is degenerate, but UUU will always code for phenylalanine. You A particular codon will always represent that and that amino acid only. For example, UUC will always code for phenylalanine. So this is basically unambiguous, which means a particular codon will always code for that particular amino acid. It is, genetic code is universal. What does universal means? Which means that UUU will not only code for phenylalanine in bacteria, but also in human beings. So everywhere UUU will code for phenylalanine. Okay, so this is the universality of genetic code. It is non-punctuated, which means UUU, UAC, UA, G. So basically, the reading of genetic code is non-punctuated. So once the first codon gets over, the next codon is there, then the third codon is there, so on and so forth. So genetic code is non-punctuated. And finally, non-overlapping. Okay, non-overlapping means it's always the first three, then the next three. It's never like U and then UAC. So these three is there, and again this three is taken, and then this three is taken. It's not like that. These three, and then non-overlapping sequence, one after other. So these are the features of genetic code. Again, hope you understood the difference between non-punctuated and non-overlapping. Non-punctuated means there is no break in it, and non-overlapping means there is no overlapping sequence in it. So these are the important characteristic of genetic code. Now, again, let's look at some of these properties one by one. So first is genetic code is degenerate or redundant, which means, for example, here you can say UUU and UUC both code for phenylalanine. Okay. Or for example, CUU and CUC both code for leucine. Even CUA and CUG also codes for leucine. So more than one amino acid is represent. One codon can more than one codon can represent a single amino acid. That is called as degeneracy. Now, what is the property in degeneracy? You have to remember that degeneracy generally lies in the third base, which means if you see both of these code for phenylalanine, and the degeneracy is in the third codon, which means the first two codon is same. The third codon, here you can see in the first case it's U, and in the second case it's C. So the third base is where the degeneracy will lie. Okay. Next is, for example, AGA and AGG both code for arginine, but the difference is in the last third codon. There are some very important points about the degeneracy. For example, amino acid which is represented by a single codon, both UGG and AUG. UGG represent tryptophan and AUG represent methionine. So these are the only two amino acid which has no redundancy, which means methionine will always be coded by AUG and tryptophan will always be coded by UGG. So these two amino acid has no redundancy. Rest all the other amino acids will have redundancy. The next question is, there are three amino acids which have maximum number of codons, that is 6 codon. Serine, leucine, and arginine. These three have the maximum amount of redundancy because all of these three are represented by 6 codons. So this is about the redundancy of genetic code. Then obviously unambiguous, I have told you that one codon can represent only one specific amino acid. For example, AUG will always represent methionine, or UUU will always represent phenylalanine. Genetic code is universal. Universality means all across a species, from bacteria to human, one codon will represent the same amino acid. For example, AUG will represent methionine even in a bacteria and us, or UUU will represent phenylalanine even in a bacteria or us. Now, there is an exception basically. The exception to this genetic code being universal is mitochondrial DNA. I mean, mitochondrial DNA, UGA is a stop codon. Uh, Normally in our nuclear DNA, but in mitochondria it codes for tryptophan. Again, this is a very frequently asked question. The next property of your genetic code is something called as wobbling phenomenon, and this is very, very important. Now we have seen that there are only 31 tRNA. Okay, we have seen in the cytoplasm there is only 131 tRNA, but how many codons we have got? We have got 61 codons. So obviously more than one tRNA will pair up with the same codon. Okay, if there is total 61 codons and only 31 tRNA, so more than one tRNA will pair with the same codon. So this is basically called as wobbling phenomenon. So base pairing of the third base in the codon and the anticodon is not strictly followed. So let me give you an example. Let me give you an example. For example, arginine has UCU in the tRNA. Okay, in the tRNA of arginine, you will have UCU. Okay, and this can bind with both AGA and AGG. Both can code for arginine. Okay, so what happens? Arginine and the tRNA will have UCU. Okay, and this UCU in the tRNA can bind both with AGA. This is the normal pairing. Okay, UCU will pair with AGA, but here this is the wobbling pairing. AGG will also pair with UCU. Okay, so if you see, U should not have paired with G, the third base in the mRNA. So the third base in the mRNA, the pairing is wobbling. Is this clear? Let me give another example so that so here this is the tRNA. Both GAG, GAG will have leucine. Okay, and this GAG can pair with CUC, which is the normal pairing, and this can GAG carrying leucine can pair with CUU also. This is called as wobbling pairing. So this is how 31 tRNA will be able to carry your 61 amino acid. So what is wobbling phenomenon? So this is a phenomenon. This is a feature of basically tRNA. Okay, the base pairing of the third codon of the mRNA. Remember, it is the third codon of the mRNA from 5' to 3', not the third codon of the tRNA. Base pairing of the third codon of the mRNA and the anticodon on tRNA is not strictly regulated. So what are the wobbling combinations present? So A and U on the third codon will not show any wobbling phenomenon. Okay, in the anticodon, but U can combine with both A and G. G can combine both with C and U, and if there is hypoxanthine, okay, in the tRNA at the 5' end, then it can combine with both A, C and U. Okay, so these are the wobbling base pair combination. A and C does not have any way of wobbling. So if A and C is there on the in the 5' end, it will have no wobbling. But if U is there, it will be AG. If G is there, it will be CU. And if I hypoxanthine is there, it can be either A, C, and U. So these are the base pair.